Good morning. Thank you. It is plenty cold. I guess coldest day of the year so far, something like that. So I thought I'd do this whiteboard style. I've done like the slides and the talk and all that kind of stuff. But I think for teaching purposes, whiteboard style can be a little bit more effective. And I also think that it can make it a little bit more interactive. So you're all here to learn about rare variant analysis. So who here has sequence data that they've been working with? Who's working with sequence data? So that's about, well, 2/3. That's gotten a lot more common. You asked that question like 5 or 6 years ago, and maybe like 5 people sheepishly raised their hands as like the early adopters. And now we're in this space where lots of people have sequencing data. So who's sequencing families and trios? Families, trios. Okay, case control, less common. Big samples, so more than, say, 1,000 cases or people. 100 cases, something like that, if your sample size is a little more modest. Yeah, okay, something in that, tens or hundreds, something like that. And I think that's actually, so we got this nice diverse representation of how many people we're looking at, and this nice diverse representation of how we maybe even think about sequencing studies more generally. So I'm going to talk about 2 main paradigms for sequencing. So one is this case control. So this is our workhorse of most common variant genetic studies are all in this case control universe. And the other paradigm that I'm going to talk about is trios/families. And these 2 approaches come with 2 very different sets of analytic considerations. Now, this talk is about rare variants and kind of sequencing studies and how we analyze rare variants. What do you think is rare? 1%? 10th of a percent? No, 1% is not rare anymore. Okay, 10th of a percent? Less than that? Yeah, no. One in a million, 1 in 10 million, 1 in 100 million. Maybe the only variant you see on Earth. That might be a little difficult to test as a way of thinking about it. So there's no agreed upon definition of rare variation. That's probably the first and most important consideration, is that when we're talking about rare variation, it's actually a kind of dynamic sliding scale, particularly for case control study designs. We're less interested in thinking about rare variation when it's like, a 1% allele, because we can capture that on a GWAS. Really, the figure of merit that you should think about is not the frequency, but the number of alleles, or something that we often refer to as the allele count. And the reason that the number of alleles is what matters more than, say, the frequency is that the number of alleles tracks with the behavior of the way our association tests work much more strongly than the frequency does. And what do I mean by that? Well, let's think about doing a kind of case control study. And if we have a case control study and we've got, say, 1,000 cases and 1,000 controls, that's great. Maybe we have 10,000, maybe we have 100,000, maybe we'll have 1 million in the not too distant future, whatever. Now, if I do this 1,000 case, 1,000 control study, and I see a variant, say, maybe 40 times, that's now got a frequency in my overall data set of really 0.1%, right? So, or 1%, right? Because that's going to be 40 over our 4,000. Those are our alleles. So 1 over 100. And that's what we would basically consider rare. And the reason that I put 40 here is that 40 is the number of copies of the allele that you need to see for your normal statistical association test to work properly. Okay? And what do I mean by like work properly? Well, what we're talking about here are something in statistics that we refer to as asymptotics. And these asymptotic properties govern the behavior of the distribution of test statistics that you can observe. So very intuitively, if I think about having, say, just 2 copies of a variant in an equally balanced case and control study design, right? So I've only got 2 copies now. I can have effectively just 3 configurations of my data. I can have 2 in cases and none in controls. I can have 1 in cases, 1 in controls, and I can have none in cases and 2 in controls. And these are the only configurations of data that are possible as a consequence of having 2 copies of that allele. And that means that when I do an association test statistic, I have a real hard time actually performing that kind of association test, right? Like the best possible p-value, if I were to use, say, a Fisher's exact test for a perfectly balanced case control design, would be effectively p-values of 0.5 and 0.5. Those are the best p-values that I'm going to get. And that's not really a p-value you can write home about, right? Like this is a kind of sad p-value. It's very difficult to write a nice paper. It's very difficult to have confidence that this 2-copy allele really matters in the context of the phenotype that I'm looking at. So how do we get around this? So what do you do if you don't have enough observations? What do you do? What do you think? Any ideas? Well, you can sequence more people. It's maybe very expensive. Another thing that you can do is maybe not look at one variant at a time, but now try and look at a set of variants, right? And so when we think about rare variant study designs, and we think about how we analyze rare variation in a case control setting, the first and most probably important thing we're going to do is select some rare variants, right? So that's our key idea. We're going to select some rare variants, and we're going to group them together. And we're going to try and analyze them all as a set, right? Pretty intuitive, makes sense. So how do you think you would want to pick a set of variants? Any ideas? No? It's too cold for the voices to work in the room? Is that what I'm understanding? So no ideas? No ideas on how we might select rare variants? Do we want to take all the rare variants in the genome and analyze them all at once? Does that sound like a good idea? Same gene. Okay, so that's fine. Good. Thank you, Laurent. So we can think about a gene. That's a natural unit of analysis. You could think about maybe if we understood in a genome context, maybe if we understood all of the functional elements that the genomics folks are identifying and the consequences of those genetic variants, maybe we would collect the rare variants that we think are influencing things like enhancers and promoters. That could be another way of thinking about doing it. But the gene is definitely the most natural unit of analysis, because at the end of the day, the most useful thing a geneticist can deliver anyone is a gene phenotype link that they are absolutely confident about, right? If you know a gene, that really helps. That really, really, really is a good contribution to the literature. It is a good contribution to science. It helps us do our downstream biology. It helps us do all of those sorts of things. But let's stick with this idea of picking a gene. All right, so we picked a gene. Do we want to include all genetic variation in that gene? You know, the synonymous, the missense, the loss of function. Does that seem like a good idea? No, there's head shaking no. So heads work, voices don't. Good. Okay, so maybe we want to subset the rare variation in a gene and focus on the things that we think are going to have some kind of biological consequence. So we can think about annotation. And when we think about annotation, we can do our typical gene model annotation, you know, the kind of putative loss of function, right? So these variants that we think are knocking out one or, you know, knocking out a copy of the gene, not making it work anymore. We can think about that. We can think about missense mutations. And then maybe we don't want to include synonymous mutations. These mutations that aren't changing the amino acid, most of them we don't expect to have much in the way of functional consequences. So maybe we want to think about these 2 classes of variation as a start. Now, if you go into the evolutionary comparative genomics literature, so this is the idea of taking a whole bunch of different species, looking at genes that are shared across different species, and asking what kinds of changes are allowed by evolution across species versus what kinds of mutations aren't, right? This is one way of thinking about classifying variation and understanding variation. There's a general estimate that this class of variation, missense mutations, things that are changing the amino acid sequence, result in something that, at least from the lens of selection, looks the same as loss of function about twice as much. So they're about 2x LOFs in here than there are in this is a premature stop codon. That's pretty straightforward. That's the sort of average statement across most genes. The problem is, this is very easy to tell. Like, we know how to read a gene sequence pretty well. Not perfectly. We can confirm it with RNA sequencing. We can do a couple of biological follow-up things to make sure that our loss-of-function mutations are really behaving like these loss-of-function mutations. But for missense mutations, all we know is that they're changing the amino acid sequence. And that means that we actually have to figure out what missense mutations we ought to select and what ones maybe we don't want to select. Okay? So that's hard, very hard. There are a lot of claims in the literature about how to do this. There are a lot of programs, at least a dozen different ways of taking missense mutations and trying to sort them into this kind of, we think this is more deleterious, we think this is more impactful. Those claims are not great. It's still a very hard problem. And you can understand why, right? At some level, every gene has its own structure, it has its own sequence of amino acids, it has all of its own little changes, and as a consequence of that, we have a much harder time figuring out the degree to which certain missense mutations are actually going to be impacting phenotype than others. But we can actually do a slightly better job at this kind of classification through another resource, okay? And another very straightforward resource to think about here is what happens if we start to actually get a much clearer idea about frequency, okay? So I talked about the way that we think about frequency with respect to the impact on the way our statistics behave, on our impact on the way that we can actually perform the analysis. But there's another way of thinking about frequency, which is it's giving us some information, maybe not complete information, but some information about how much selection does or does not really care about it. And this, in a similar way to our computational prediction programs, things like SIFT or Polyphen or any of the other things that have been wandering around the literature for probably 20, 30 years plus, this kind of piece of information is also pretty crappy, right? It doesn't actually tell us that much in and of itself. But if you start to combine information about frequency with information about predictions of what the consequences of certain mutations are, with information about comparative genomics and how different species tolerate or don't tolerate these kinds of missense mutations, you might actually start to do a slightly better job at being able to sort out what these missense mutations really are that matter and those that don't. And I'm just going to put a little marker down here and say we'll come back to that after we talk about the kind of trio and family-based design, because there's a different way of analyzing that, and the way we analyze this kind of data can help us interpret this kind of data. Okay? So now we've talked about annotation. Maybe we throw in frequency as another kind of conditional approach. But we're now already in this space of thinking about grouping variants together, about testing them as a kind of composite set. We're not in our more traditional, straightforward case control, like if you go to the intro to GWAS, it's a pretty straightforward, take an allele, it's common enough in the population, we can test it and see whether it's more common in cases versus controls. And that's all we're doing really when we do a GWAS, is we're taking these common variants and directly asking the question whether that variant is more common in cases versus controls. Here, we lose that resolution. We have to group variants together, and when we group variants together, maybe we have a strong expectation that they all move in the same direction. So here in our loss of function universe, we can think that all of these mutations are knocking out one of the two copies of the gene. As a result of knocking out one of the two copies of the gene, we can have a strong expectation that the downstream biological consequence of that kind of mutation is going to be the same. So maybe grouping that together and just setting up a little straightforward regression where this is maybe our phenotype, and now we have something like this, where this is the number of LOFs that you carry. Typically people really only carry one for most genes, since LOFs tend to be pretty rare. And then this is our test, right? It's just a simple regression. We're estimating the impact of loss of function mutations on some phenotype. You can do this for a linear regression if it's quantitative. You can do it for logistic regression if you want to do a case control kind of analysis. This is all pretty nice and straightforward. But that starts to break down as well if we start to think about missense mutations. We do not expect every single missense mutation is going to have the same biological consequence, right? So some may cause a major structural change to a protein, and the protein won't work anymore, and that might look exactly like a loss of function. But other missense mutations might maybe slightly enhance some aspects of the activity, or perhaps rather change the consequence of the downstream protein. So whatever the protein is doing, it might be doing it slightly differently or slightly more efficiently or slightly less efficiently. The problem is we don't really know. We don't have any ability to tell that right now unless we do a lot of detailed structural biochemical experiments, which are not very scalable and certainly cannot keep pace with the rate at which we're identifying these kinds of classes of mutations, right? Like these kinds of downstream experiments are not ones that are straightforward for us to do, at least at this point in time. So what do you do? What do you do when you have a circumstance where you might have a class of variation that both increases risk for some of the variants, but decreases risk for the other variants? Well, let's think about what that's going to do to our little dummy counts. So let's just take a bunch of doubleton variants. Okay, so 2 copies of the variant in the total data set. That's all we're going to talk about. And if we think about these doubleton variants, our expectation under the binomial, if we have equal numbers of cases and controls, is pretty straightforward, right? So 1 to 1, 0 to 2. So we might expect a quarter of our mutations to fall over here, a quarter of our mutations to fall over here, and half of the mutations to fall equally split between cases and controls. Okay, all very intuitive, that makes sense? Okay, so all I'm doing is saying we've got a 2-copy variant, and this 2-copy variant we're seeing in this kind of breakdown in the binomial space. So what do you think, and this is our number of counts in cases, and this is our number of counts in controls. So what do you think happens if a variant increases risk? Does it stay in these proportions? No. What happens? So it moves this way. All right, great. All right, so maybe for the risk variance, now let's say that just for the sake of argument, we have really strong acting risk variants, right? So much so that they increase risk by like 20, 50, 100-fold or something like that. Maybe we'll even call them fully penetrant just for the sake of argument. Those mutations are going to end up with this pattern, right? Pretty straightforward, nice and intuitive. Now what happens if you have a variant that doesn't increase risk but actually decreases risk? So it goes the other way, right? So now let's say that it is a fully protective genetic variant. We don't really identify these very well. They're hard to figure out, but just for the sake of argument, just for the kind of didactic purposes to get yourself thinking about it, these protective variants, they will follow this kind of pattern, right? So we're going to have this shift the other way. Now let's say that they're equally split between risk and protective. Again, just for the sake of argument, I don't think that's really going to be the way it plays out, but who knows? Genome's a big place. There are lots of genes. In that circumstance, our expected number of alleles running this kind of regression will give you absolutely no signal. They'll perfectly balance each other out. We're going to have, a bunch of variants here, a bunch of variants here, and maybe we'll have some neutral variants mixed in because we still suck at figuring out which missense mutations are actually relevant and which ones aren't. And so what we'll end up with is no relative increase or decrease of this class of variation, but we will have ended up with something else. And that's actually that we're going to see much more variability in the assignment of these rare variants across cases and controls than we would expect to see by chance. So if I calculate the variance in the distribution of assignment of alleles rather than just the mean, if I calculate that variance is going to be much higher, right? Because normally when we calculate the variance, we can think about the variance as simply defined as the kind of probability of the kind of, genotype or whatever, the genotypes times the frequency of those genotypes, minus the average, the kind of mean of the frequency of the genotypes, and we're just going to square that. And so in this circumstance, our variance is nice and straightforward. We would do 2 minus, so 1, so we're just going to do for the count of alleles in cases, it would be 2 minus 1, well, 2 times a quarter minus 1, right? Plus quantity squared, plus, 1 minus 1, because that's our average number of alleles, quantity squared, plus 0 times a quarter minus 1 quantity squared, right? So we do the frequency of those things. And actually, sorry, I want to move this out here. That's the way I want to do it. And when we do that, what we end up with is a quarter plus a quarter, so a half. That's our variance. But now what happens if we only see, let's say half the time we see this, so this will now move up to half, and we see half the time we see this, and we don't see any of this. Well, what's going to happen to the variance here is that we're going to have now a half plus a half, and our variance is going to equal 1. So you can see that the variance of the distribution of the assignment of alleles has increased because of the way that we've constructed. We have exactly the same mean, but half the time we got this and half the time we got this, and that's increased the variance in the distribution of our allele assignment. Now, this kind of testing framework, this kind of approach, where we scrutinize the variance rather than just simply the mean, this is called sometimes the C alpha test statistics, because what we're trying to detect in this circumstance is a mixture of variants that are increasing risk and decreasing risk. Okay, does this make sense? That we're actually testing that variability in the assignment of alleles rather than testing the alleles themselves in a kind of more traditional directed fashion. These kinds of tests are also what live underneath the so-called variance components tests, or things like SCAT. So what SCAT does is it basically asks how similar are people genetically, how similar are people phenotypically, and if you're more similar genetically and more similar phenotypically, then we think that variation in that gene might be relevant to the outcome. But it's doing exactly the same kind of machinery. What it's doing is it's asking this question of what is actually going on with respect to the assignment of the alleles and how much variability is there. And these kinds of tests, SCAT and C alpha, and those kinds of approaches are really the way that we deal with this fact that we have a whole bunch of uncertainty about functional prediction, about what variants might be doing in terms of increasing or decreasing risk for disease. Does that all make sense? So that's just kind of introduction to how we think about rare variant testing in the case control setting. In the trio and family setting, we actually have some very different considerations. Now, one set of considerations are very simply, the kinds of linkage analyses that really dominated genetics in the 80s and 90s and led to the mapping of a whole bunch of genes, including Huntington's and cystic fibrosis. That's a lot of how we think about rare variant analysis in larger families. But let's talk about trios for a moment, because I think the trio paradigm was really transformed, particularly the identification of very strong-acting mutations that are effectively reproductively lethal, okay? That doesn't mean that they kill the person. It just means that the person is extremely unlikely to have any children. Okay, now for another little thought experiment. What do you think the frequency of a variant is if it renders you completely sterile? So there's one variant, and it removes your ability to have children altogether. What do you think the frequency of that is in the population? What's that? It shouldn't stay in the population, right? So it's not going to get transmitted in the population. So if we see it in the population, how did it get there? Mutation rate, exactly. Okay, so this is like a really key idea in population genetics, this idea of when you have mutations that are under extremely strong selective pressure, the expectation for the frequency of that variant is equal to the mutation rate divided by S. And this S, this idea of S, is the selection coefficient against that class of variant. So basically the reduction the relative reduction in the number of children that you expect to have, conditional on carrying that variant in the population, okay? Now, S is a funny thing. S is like relative, it's not absolute, but because most variation is basically neutral, you can think about it as relative and it works out okay and it's all fine. But if you think about those variants where, you know, what I just described for you to get you thinking about this was an S of 1. This idea that if you have that variant, it's not in the population, and therefore the frequency is simply what the mutation rate is. So when S equals 1, that's like reproductive lethality, we have the mutation rate. Now the base mutation rate in the population is approximately, this is somewhat contested, but approximately 1.2 e minus 8 for point mutations. So at a kind of given point in the genome, we expect a mutation rate somewhere in that neighborhood. There's like a range, different estimates from different things that you look at. The catch with this mutation rate, though, is that it actually varies as a function of the local sequence context. So there are a couple of papers that talk about thinking now not whether we're going to go from A to C, et cetera, but if we now have a trinucleotide context, so maybe we have the base before, the base that we're interested in interrogating, and the base after. This base has a different mutation rate conditional on what's around it. So this motif has a higher mutation rate than if the motif was like this. So that is to say that if the 3 bases, the C here has a lower mutation rate than the C here. And this is just part of the way mutation arises in the population. There's some funny biochemistry that people have worked on for a very long time to demonstrate that. There are other forces that influence this above and beyond the base before and after. There are some arguments that maybe taking an additional base on either side might improve our characterization and estimation of the mutation rate. There's also methylation, so the degree to which the genome is methylated influences the degree to which these kinds of mutations arise. And the reason that I'm talking to you about these different kinds of mutation rates is because the real power of the trio paradigm is to look at those mutations that are newly arising in the population and ask whether they tend to cluster in a gene. So if I got 10 patients of some phenotype that we define, and I'm only looking at those 10 patients, and they all have a de novo mutation in the same gene, that would make you think that that gene is likely involved in that disease, right? Like, you know, that feels like a pretty good result, right? We'd get excited about that. Now, if I say that we've got 10 patients and we see 5 de novo mutations in that gene, we're probably still pretty excited, right? Like 5 mutations seems like a kind of unusual thing in a single gene. I've told you that the base mutation rate's pretty low. If they're all, say, loss of function mutations, the average loss of function aggregate mutation rate for a gene is on the order of something like 1e minus 6, something in that kind of territory. That's, you know, much, much lower. That's like a pretty rare phenomenon, right? So I've got 5 mutations out of my 10 patients. Now, when I get to 1 mutation, what do you think? Do you want to believe it? No? No, you don't want to believe it. So 1 mutation you don't want to believe. Do you want to believe 2 mutations? No, maybe. I bet if I'd started at like 3, I'll be much more enthusiastic about 2. And the reason that you can't give me an answer is because you don't have a model. And the way that we think about testing trios and de novo mutations is we build a model of this mutation rate, and we're going to use it in a couple of different ways. So one of the ways that we're going to use this model for mutation rate is to calculate how many de novo mutations would I expect to see in a given gene of a given variant class, conditional on my sample size, right? So you just build this model. So you know what the mutation rate is for every single base. You can then say, or you can like predict that pretty well, or model it pretty well. You then say, here's my overall de novo rate, and so I might have now this mu of LOF of, say, you know, PCSK9, whatever, it doesn't really matter, pick your favorite gene. And now I know what my mutation rate is for this class of variation. And what I'm going to do is I'm going to model this as something we refer to as a Poisson process. So the Poisson distribution is a distribution that is extremely well suited to dealing with count data that is, discrete, so discrete count data, 1, 2, 3, et cetera, and relatively speaking, rare. So when the Poisson gets big enough, it turns into a normal distribution. Just use normal distribution. Everyone loves normal distribution. Works a lot better. But the Poisson does a really good job of figuring out what the kind of distribution of effectively random events are. And each de novo mutation that we're talking about is that kind of random event. And the rate parameter for the Poisson, that conditions both the mean and the variance of the Poisson, that rate parameter is very simply the mutation rate, and we know what the mutation rate is. And so we can directly test whether the rate of mutations that we're observing in our data is a lot higher than the rate of mutations that we would expect to see. And for Poissons that have pretty, so the kind of how many loss of function mutations do you expect to see in, a kind of given gene with 10 people, it's going to be pretty small. And so even when we see 2 observations, that actually becomes extremely unlikely because the Poisson ends up looking a lot like this and then like that, where we have almost all of the time we would expect to see no mutations, maybe a little bit of the time we'd expect to see 1 mutation, and then extremely rare to see greater than 1 mutation. And that's how we can take 2 de novo mutations in a set of, say, 100 trios and say, hey, 2 de novo mutations that are a loss of function, that's a really big deal. This is not the same as saying, I see a de novo mutation in this patient with this disease, and therefore I believe this gene. That is actually something we actively have to move away from because a single observation is bound to happen given a sufficiently large sample size. If I keep sequencing trios, I'm going to see a background rate of these mutations because they occur in the general population at some rate. And the rate's about 3% or so of individuals in the population will have a de novo mutation in that's loss of function in a gene. So in this room, someone probably has a de novo loss of function mutation. Thereabouts. Not pretty likely, actually. And that's a funny thing to think about, but it is something that, most of those aren't actually going to do much of anything with respect to any given phenotype because we don't expect every gene to have massive phenotypic consequences that exist in the genome. And so the way we analyze these kinds of trio data sets is, again, we build this model of mutation rate. We use that mutation rate to calibrate an expected number of de novo events. We have our observed de novo events, and we use a kind of Poisson regression style model to see whether our model fits our reality, and that can give us an actual gene-wise p-value. We get a p-value for every gene in this kind of circumstance. Now, the other way that we use this mutation rate, this is a nice important kind of corollary, is to go back to this question of how do we think about missense mutations, and actually how do we think about genes? Do we think that there are genes that are more or less subject to purifying selection in the population? And the answer is yes, there are genes that are more or less subject to purifying selection in the population. Some genes are really critical. If you knock out a copy of that gene early in development, then it's really bad. Whereas there are a whole bunch of other genes where you knock out that gene early in development and it really does nothing, right? So you have an olfactory receptor, you got a new loss of function mutation in an olfactory receptor. Okay, it doesn't matter. We see lots of those mutations. Actually, we see lots of loss of function mutations in olfactory receptors in the population. And that's selection telling us in another way that those genes aren't so critical for, say, reproductive fitness, for things that natural selection prefers to think about. Okay, so let's talk about how we do that. So the way we really focus on this is we count the number of variants that are, say, synonymous that we observe in a very large reference data set. So something like NOMAD or EXAC, you can think about these large reference data sets. We count the number of variants in a gene that are synonymous, and we model basically what fraction of these variants we're observing as a function of the mutation rate. So we build this model to predict this. So this is what we're predicting. We're going to predict this based on the base level mutation rate. Okay? And that actually does a remarkably good job of capturing the variability in what, how many synonymous mutations we observe in a given gene. We actually capture like 95% of the variability in the number of synonymous variants in a gene based on using just the mutation rate for every possible synonymous mutation in that gene overall. And that works really well. When we take the parameters that we get out of this model and then apply them for loss of function and for missense, what we see for every gene, so this is like, here's our expectation, here's our observation, and then for synonymous variants, we see this bubble, everything works pretty well, it's really good. I don't know if that's super easy to see, maybe I'll draw what we see over here. So and I'll finish here. So for synonymous variants, we see basically a cloud that looks like this. So our expectation that's conditional on the mutation rate versus the observation of the number of variants. Okay, so we basically run just a pretty simple regression model to get at this answer. When we apply this to missense mutations, we see this kind of pattern. So we actually see a kind of, and then even some of this sort of crops out. So what we see is that genes pretty much in general have a reduction in the amount of missense mutation compared to what we would expect. Some genes, missense mutations don't really matter, selection doesn't really care, or there are other population genetic forces that we're not actually modeling that are confounding our analysis of the essentiality of given genes. So some are behaving just fine, but there's a whole chunk that are reduced in variation, and it's a kind of continuous drop-off. Like, that there are some genes that lose a lot of missense mutations that are living more down here, and then there are some genes that are losing some missense mutations that are living over here. And then when we look at loss of function, what do you think we see when we see loss of function? Even lower, and that's right. That's exactly right. So we do see a handful of genes where loss of function mutations are basically unperturbed. But then we see a much larger chunk where we observe few, if any, loss of function mutations, even when our model says we should be observing plenty. And we can take this kind of reduction and quantify this reduction in loss of function mutations based on the mutation rate. Again, it all comes back to that mutation rate. It's really important for interpreting genetic variation. So based on that mutation rate, we can then assign now a continuous score about the relative depletion of loss of function mutations at a gene level. And this has been the workhorse for something referred to as PLI, the probability of loss of function intolerance. We also refer to this as constraint. In the recently released Nomad paper on bioRxiv that came out Tuesday on bioRxiv, when Conrad posted it, he introduces something that he's referring to as LUF, which an eggy name, I suppose, which is the upper bound of this observed rate of mutations over the expected rate of mutations in a gene level that's now attempting to get at this idea of a kind of continuous measure of the degree of natural selection. Because what we're really interested in doing is identifying those genes that are under the strongest amount of purifying selection more generally. And Hillary's come in, which I'll take it as my cue to just wrap up and say one more thing. This is not the only way that we can start to leverage large-scale population references to understand what genes matter and what genes don't. There's some work from Slava Petrovsky and David Goldstein that rather than using the amount of variation that you see versus what you would expect to see, looks at the relative rate of functional mutations above some frequency threshold to the rate of those mutations below a frequency threshold, right? And so this is called RVIS, and that is now taking all the variation that you see under the assumption if all you're seeing are common synonymous mutations and rare missense and loss of function mutations, that's consistent with the idea that those common missense mutations are just missing. And as a result, that's another way of getting at this way of sorting out what genes are important and how we start to interpret them. And with that, I'll say thank you, and I guess invite the crowds at the gates to come on in and wrap up.